Why is there still mitochondrial DNA? Mitochondria is considered the energy generator for the cell, and the introduction of mitochondria in eukaryotic cells gave rise to multicellular life. Through aerobic respiration, mitochondria uses oxygen to create ATP, or adenosine triphosphate, an essential molecule for transporting chemical energy that is used in cellular processes. Billions of years ago, an alpha proteobacterium was consumed by another cell. Mitochondria have their own circular DNA that is very similar to the circular chromosomes of bacterial DNA, which is evidence for the endosymbiont theory of the origin of mitochondria. Predecessors to the present-day mitochondria have thousands of genes. Now they only have a select few. Since then, most of the mitochondrial genes have been transferred to the nucleus or lost. The purpose of this scientific research is to see why so many genes in the mitochondria are lost and why there are select few still there. A possible reason genes have transferred to the nucleus is to avoid mitochondrial mutagens and Mueller's ratchet, which is a buildup of harmful mutations that can't be reversed. For the select few genes that stayed in the mitochondria, some alternative hypotheses propose that those genes will optimize the performance and allow for a better localized control over the organelle. To test their hypotheses, Ian Johnson and Ben Williams analyzed the evolution of the gene loss in the mitochondria using an algorithm, hypertraps. While this research leads us one step closer to solving the mystery of mitochondrial gene loss, the results are only hypothetical, since it is taking data from the past to predict gene loss patterns.
The algorithm hypertraps was constructed by Johnston and Williams to calculate the most probable and preferential trajectories of the evolutionary dynamics of the mitochondria organelle. To generate data with hypertraps, the researchers used the monotypic genus of the Jacobid eukaryotes due to the organism's vast mitochondrial genome. It was found that across the 2015 species tested, 74 distinct combinations of genes were present in visible clusters. The results show that the evolutionary history of mitochondrial gene loss in a typical mitochondrial genome was remarkably structured and non-random. Although different lineages experience different gene loss trajectories, the overarching trend holds for eukaryotes. Gene loss among the phylogeny was characterized into early lost, intermediates that are present in plants and fungi, and highly retained genes. It makes sense that many genes would have been transferred to the nucleus. Why would you want to store your pictures in different places when you could keep them all in a sturdy, centralized location?
While this sounds logical, these highly retained genes are vital to the organelle functioning and have allowed the mitochondria to stay independent of the nucleus. The genes that Johnston and Williams identified as most likely to be retained were ones related to the guanine cytosine content of the DNA in question and the hydrophobicity of the proteins created by the mtDNA. The data suggested that these genes are conserved in the mitochondrial genome to allow for localized maintenance and control of gene expression. More specifically, if a stress or demand is inflicted to an individual mitochondria, that organelle has the ability to adjust its electron transport chain complex without affecting the performance of other mitochondria in the cell, which would happen if these genes were absorbed into the nucleus. The mitochondrial DNA in these genes utilizes an ancient pattern that allows DNA to strongly bond together and resist breaking apart. In the production of ATP, high-energy free radicals such as nitric oxide are emitted. Free radicals are missing an electron in their outer shell, which causes them to seek out a molecule to steal an electron from. Essentially, a free radical is like an energy robber. The researchers believe that genes that are able to endure this harsh environment are the ones localized in the mitochondria today.
The guanine cytosine content and the hydrophobicity of the proteins created by the mitochondrial DNA serve as the backbone of Johnston and Williams' research, as these traits closely match the pattern of gene retention throughout the eukarya phylogeny. The results of Johnston and Williams' study have shed light on a mystery that has stumped scientists for decades by relating the evolutionary phenomenon to the preservation of key genes that prevent mitochondrial damage. Understanding why the mtDNA has retained specific genes helps to define the selective pressures placed on mitochondria in the past and the evolutionary paths that their genomes might take in the future. This research paves the way for new discoveries in synthetic biology and the opportunity to apply Hypertrap's algorithm even further to the chloroplast and mitochondrial diseases.